The overall goal of this procedure is to reproducibly generate human pluripotent stem cell-derived cardiomyocytes in size-controlled cell aggregates.
This method can help answer key questions in the stem cell field, such as how human embryonic stem cell aggregate size and geometry affect endogenous interactions between differentiating cell types that affect cardiac induction efficiency. The main advantage of this technique is that we eliminate a source of variability from conventional stem cell differentiation protocols, aggregate size, to efficiently and consistently produce cardiomyocytes.
Though this method can provide insight into how aggregate size can influence cardiac differentiation, it can also be applied to other systems, such as neural or endodermal differentiation from human pluripotent stem cells.
First, centrifuge a single-cell human pluripotent stem cell, or hPSC suspension, at 200 times G for 5 minutes. Following the centrifugation, aspirate the wash medium and resuspend the cells in aggregation medium at a density of 1.2 times 10 to the 6th cells per milliliter. Next, seed 1 milliliter of the cell suspension to each well on the prepared microwell plate and evenly distribute the cells. To seed a large number of wells, periodically vortex the cell suspension to prevent settling. After seeding, centrifuge the plate at 200 times G for 5 minutes. After the spin, observe the plate under the microscope to confirm cells have spun to the bottom of each microwell. Then, incubate the plate for 24 hours at 37 degrees Celsius in a 5% CO2, 5% O2 hypoxic incubator.